The ER, Golgi apparatus, and their products, such as vesicles and lysosomes, form the endomembrane system. Let's examine the synthesis of a glycoprotein found in mucus. We'll view this process from the DNA instructions to export out of the cell. Segments of DNA in the nucleus called genes contain the instructions to produce particular proteins. The DNA instructions are copied into an RNA molecule. Messenger RNA, or mRNA for short, exits the nucleus via the nuclear pores. The RNA instructions bind to a ribosome in the cytosol. A signal sequence is then synthesized by the ribosome, and this signal binds to a signal recognition particle, or SRP, in the cytosol. The SRP then binds to an SRP receptor protein embedded in the membrane of the endoplasmic reticulum. This activates the ribosome, and it synthesizes the rest of the protein per the RNA instructions. The ribosome, RNA, and SRP disassemble when protein synthesis is complete. Inside the lumen, chaperone proteins ensure the ER protein folds into its proper three-dimensional shape. Also within the lumen of the ER, enzymes will add a carbohydrate to the protein to form a glycoprotein. This process is called glycosylation. A vesicle containing the glycoprotein buds off the ER, travels to the Golgi, and emerges with the cis face of the Golgi. Within the lumen of the Golgi, enzymes modify the glycoprotein. The modified glycoprotein buds off the trans face of the Golgi and releases the glycoprotein outside the cell. This process of a vesicle merging with the cell membrane to export materials out of the cell is called exocytosis. Remember, the ER, Golgi, and vesicles are all part of the endomembrane system. Vacuoles act as storage compartments within cells. The most prominent vacuole is the central vacuole in plants. It is a storage organelle for proteins, noxious substances to deter herbivores, water, food, salts, pigments, and waste. Central vacuoles also contain hydrolytic enzymes to break down waste and other cell components. The vacuole is hypertonic to the rest of the cell, and this osmotic pressure allows plant cells to be turgid. 50 to 80% of the cell may be taken up by the central vacuole. Mitochondria are the site of aerobic respiration, an oxygen-requiring process that converts chemical energy to ATP. Mitochondria are found in all eukaryotic cells. Here are the mitochondria. A single one is called a mitochondrion. Mitochondria are easy to spot. Each mitochondrion is enclosed in a double membrane. The intermembrane space lies between the outer and inner membranes. The inner membrane folds inward, producing sac-like structures called cristae. The matrix is the area within the inner membrane. This diagram shows the locations of the outer and inner membranes, the intermembrane compartment, and the sac-like cristae. Make sure you can locate those structures on this micrograph of a mitochondrion. The chloroplast is the site of photosynthesis and can be found in plant and some protist cells. Here are the chloroplasts. The chloroplast also has a double membrane. A space called the stroma is found within the inner membrane. Found in the stroma are thylakoids. Thylakoids are membrane pouches that contain chlorophyll, the molecule that captures light energy. The thylakoids are stacked on top of each other to form grana. This diagram shows the locations of the outer and inner membranes, the stroma space, and the thylakoids. And there are plastids other than chloroplasts in the plant cells. These cells are from a potato and contain amyloplasts, which store starch. The cells have been stained with iodine so that the starch can be better visualized. It is hypothesized that mitochondria and chloroplast originated by endosymbiosis, in which a larger cell engulfed, but did not digest, a smaller cell in the past. The evidence? Well, both organelles are similar to bacteria in structure and have their own circular chromosomes. The last structure we will discuss are the elements of the cytoskeleton. The cytoskeleton is composed of protein filaments. These filaments determine the shape of the cell and can allow some cells to move. They also act as a highway for organelle movement and are crucial during the division of cells. The three main types of protein filaments we will examine are microtubules, microfilaments, and intermediate filaments. Microtubules are composed of two different types of protein subunits, alpha and beta tubulin. The alpha and beta tubulin bind to form dimers. These dimers polymerize to form a hollow tube. Microtubules are the thickest of the cytoskeletal elements and are produced in a structure called the microtubule organizing center. They have many functions in the cell. They assist in transport within the cell, are critical to cell division, and are found in structures that allow the entire cell to move. While plants have many microtubule organizing centers, most animal cells only contain one, the centrosome. A pair of microtubule bundles called centrioles are found in the centrosome. The chromosomes are stained red in these plant cells. The threads you see between the chromosomes are microtubules that ensure the DNA is properly distributed in dividing cells. Axonemes are structures that extend from the surface of the cell and are composed of microtubules in a 9+2 arrangement. They attach to the cell at the basal body, which is almost identical in composition to a centriole. The 9+2 arrangement of the axoneme refers to the 9 microtubule doublets that surround the two central microtubules. Here's a diagram of a cross-section of an axoneme. The whole structure is surrounded by the plasma membrane. All the microtubules are linked together, and a motor protein called dynin is found between the doublets. When the dynin is phosphorylated, it changes shape, and this causes the microtubules to bend. The bending of the microtubules results in a swimming movement. Short hair-like axonemes are called cilia, while longer whip-like axonemes are called flagella. This paramecium has many cilia that bend back and forth to allow the organism to move. And these sperm have only one flagella that allow them to swim to the egg. Microtubules also act as highways that motor proteins walk on to transport vesicles in the cell. Let's see how that is done. Here's a vesicle with a motor protein called kinesin attached, and here's the microtubule. Since it is composed of two different types of tubulin, the dimers are arranged in a specific pattern. This gives the microtubule different ends, the plus end and the minus end. When ATP phosphorylates kinesin, the kinesin changes shape, rotating one head of the kinesin towards the plus end of the microtubule. And each time the kinesin is phosphorylated, the other head is rotated, moving the kinesin with its vesicle cargo along the microtubules. Here we see a video of vesicles being transported along microtubules. It is hypothesized that the kinesin takes about 100 steps in a second. Microfilaments are composed of protein subunits called actin. Two strands of actin are twisted together to form the filament. Microfilaments are the thinnest filament, yet the twisted arrangement of the strands allow them to be strong and flexible. They maintain cell shape by resisting pulling forces, permit cells to change shape, and move organelles in the cell. The microfilaments have been labeled with a green fluorescent dye in this cell. The net-like arrangement helps strengthen the cell and define its shape. Microfilaments can be found interacting with another protein called myosin to allow for different types of movements. Both are found in some cells, like muscle cells, and allow the cell to contract or shorten. This pair pinches animal cells in two during cell division, a process called cytokinesis. And myosin and actin also work together to move the contents of cells in plants during cytoplasmic streaming. Intermediate filaments can be composed of various proteins. Two segments form a dimer, and these dimers are twisted to form a coiled filament. Intermediate filaments are between microfilaments and microtubules in size. They maintain the shapes of cells, anchor organelles in place, and form the nuclear lamina underneath the nuclear envelope to maintain the shape of the nucleus. Intermediate filaments are stained with green fluorescent dye in this micrograph. In addition to defining the shape of the nucleus, they also anchor the chromosomes to the nuclear envelope.